Good morning, everyone. Well, still people come into the meeting. This meeting will be recorded, I hope, and did Lorraine agree to that? Okay, without further ado, it's my pleasure to welcome everyone to our monthly invited seminar series. Today we have a very prominent pioneer in nutritional metabolomics, Professor Lorraine Brennan. She is at UCD, which is not UC Davis. So that's it, because at UCD is for us, it's always like Davis, of course. Right? But no, she's in Dublin, Ireland, and at the Food and Health Conway Institute, she was awarded an ERC Consolidated grant in the field for discovery work, and she is also involved in 2 European consortia. She looks at metabolomic profiles in food intake and really tries to pave the way for using metabolomics as discovery tool for biomarkers of food intake towards strategies delivering personalized nutrition. I came just from another talk where people said, it's actually Peter Dorrestein who said he has some evidence saying that soda intake increases or accelerates Alzheimer's decline. So, from his data so that's like the idea of precision nutrition, to tell people like, okay, you better slow down on this or make more of that, based on data, not just assumptions. She has served as director of the European Nutrigenomics Organization for 5 years. And she led a number of important initiatives, including the development of an early career network, expansion of membership of the organization. She was appointed recently to the National Academies of Science, Engineering, and Medicine Standing Committee on Evidence Synthesis and Communication in Diets and Chronic Disease Relationships. So she published a lot of papers, and we're really happy to have you with us today. Lorraine, it's a pleasure to meet you. Looking forward to that. Just to tell you how it works, we invite everyone who comes into the room to post questions while you talk into the chat room, and then Dr. Arpana Vinaya will lead the discussion afterwards, kind of like bundling those and then posing those questions to you, Lorraine. Okay?
Okay, very good, very good.
That's how we do it. Okay, Lorraine, the floor is yours.
Thank you very much, Oliver. I'll just start maybe by just trying to get my screen share working here for a moment. And if you could tell me you can see that, that would be great.
Yes, we can see that.
You can see that. Okay, great.
Yes.
I never quite know whether it's working properly or not. So thank you very much. It's an absolute pleasure to be speaking to your UCD today. As Oliver said, I'm also from UCD. We also think that our UCD is the only UCD. And our UCD stands for University College Dublin. So I'm based in Ireland. And as Oliver said, I'm based in the Institute of Food and Health and the Conway Institute. And I lead a research team that's interested in metabolomics and using it in nutrition research. So what I'm going to do today is really talk to you about metabolomics in nutrition. And I'm going to specifically focus on 2 main parts that we've been very interested in recent years. And the first one is using food intake biomarkers, and these biomarkers are discovered using metabolomics, and then how we can actually use them to determine intake of certain foods and actually also to look at dietary patterns. And the second aspect I'm going to talk about is the concept of precision or personalized nutrition. So to get started then, metabolomics has played a huge role in nutrition research over the last 10 years, and in particular over the last 5 years. And really the synergies between nutrition and metabolomics come from the fact that the food that we eat contains thousands and thousands of small molecules, and we can measure these, of course, using NMR and LCMS. And these molecules give us information about what people are eating. But also the food that we eat has huge impacts on our metabolic pathways. And by looking at these metabolic pathways, we can understand more about the impact of food on our health. So if you broadly look at what metabolomics has done to nutrition research over the last 5 to 10 years, you can define the areas really broadly in these 4 areas. The first one, as I said, is looking at biomarkers of food intake. And I'm going to touch on these in detail during this talk. The second one is this concept of using metabolic profiles or metabolic phenotyping to help precision nutrition. And this is this whole concept of moving away from one-size-fits-all guidelines to more tailored recommendations. And the third one is really understanding the impacts of diets and the molecular underpinnings of the health benefits of diets. And this is really important. And this is really important for us to gain substantial evidence so that we can promote, or we can really have scientific evidence behind the promotion of certain foods. And the fourth one then is in the area of study of diet-related diseases. And as I said, I'm going to focus really on these first 2 applications in today's talk. So then, just to get started on food intake biomarkers, as I said, these are really important. And I'll take a step back and I'll explain from a nutrition perspective why they're so important, because they really are important from our perspective. And really the importance comes from the fact that our current methods of measuring intake, such as food diaries, food frequency questionnaires, and 24-hour recalls, are all relying on self-reported data and all have well-established limitations associated with them. And examples of some of those limitations are that people just find it difficult to remember what they've eaten. So their recall of what people have eaten is very difficult. And in some populations, it's even more difficult. People will often over-report foods that they perceive to be healthy and under-report foods that they perceive to be unhealthy. In the case of food diaries, where you're working with a food diary and you're recording the information as you go throughout the day, in this instance, people will also often alter their behavior or their food because they know they have to report that food intake. So as a result, we do have huge issues with our self-reported data. That doesn't mean that they're not useful in any means. They are useful, but we do have limitations with them. And where the biomarkers come into play is that these biomarkers can help us overcome some of these limitations. They have the potential of offering objective measures of intake. Now, I will say from the outset that I don't believe that biomarkers will replace completely these self-reported methods, because there are just too many different types of foods that we want to measure, and there are complexities associated with food intake that we need to know about. So was the food eaten as part of a meal? Was it eaten as a meal? Was it eaten as a snack? So these are all important social aspects that are important to know as well. But where the power of it all will come into play will be in joining this self-reported data with some of the biomarker data that we can get from metabolomic experiments. So we're particularly interested in developing these biomarkers and in trying to use these biomarkers to help improve the accuracy at which we assess what people are eating. So these biomarkers of intake food intake biomarkers can be used in a wide range of applications. And I'm just going to touch a little bit on some of them here. So over here on the left, we can use biomarkers once we have good biomarkers to measure adherence to dietary interventions. And this is really, really important when we're trying to demonstrate the efficacy of an intervention. So from a human nutrition perspective, when we do randomized controlled trials, what we often get criticized about is that we don't know anything about the compliance of our volunteers that were involved in that study. And this is where biomarkers of specific foods can help us hugely in that. And it can raise then the level of certainty we can have on the outcomes of our studies. And the second one is in relation to objective measures of dietary intake. And this, of course, is the holy grail. This is where we want to be. We want to take a biomarker and we want to know exactly what somebody has eaten using that biomarker. The third one is an area that we're also interested in, and this is the concept that you use these biomarkers to overcome the limitations that are associated with self-reported data. In other words, to correct self-reported data for some of the errors that are there. And finally, then you can use these biomarkers to study the relationships in health and disease parameters. So you may have a marker that's related to food intake, and then in a cohort study, you can use that marker to study the relationship between the dietary intake and a health outcome. So then how do we actually discover these biomarkers, and what's our protocol for doing it? So we and others in Europe, and in particular a consortium called the Food Bowl, which is funded through the Giant Programming Initiative, have been following this approach here. And this is where we perform controlled intervention studies using exact intakes of certain foods. We feed the volunteers these foods, and we collect biological samples following the consumption of the foods. We take samples, we run our metabolomic analysis of them, we identify metabolites that changed following consumption of the food, and then we have our putative biomarkers of intake. We then perform a dose response study where we feed volunteers different quantities of the food, and we look to see, do these biomarkers increase with increasing intake of the food. And then finally, it's really important that we go into larger independent cross-sectional studies where people are usually eating a variety of different foods and a variety of different diets, and we look to see, do our biomarkers hold up in those instances. So I'm going to take you through some of these concepts over the next few slides. So just as an example, I'm going to take one study that we done a number of years ago, looking for biomarkers of legume intake, and this was done in the context of this particular project that I mentioned earlier, the Food Bowl project. So in this project, we fed volunteers different foods in 4 particular foods, and for here, I'm particularly interested in the pea intake, so pea being our legumes in this instance. So we fed our volunteers these different foods on different days. We collected postprandial samples, so samples at 2, 4, 6, and 24 hours following consumption. And we analyzed all of the urine samples then using LCMS. We done our usual feature extraction and alignment of our alignment and feature extraction of our data, and then ended up with our data to bring it into our statistical analysis. And then just to give you a flavor of some of the data that we've obtained, this just shows you here, these are samples prior consumption of the peas, and these are samples post-consumption of the peas, and then we can identify biomarkers that are changing following consumption of the legumes. We went on then to identify some of these features, and you can see here, these are some of the excretion kinetics from these metabolites. In green are the legumes, and in orange are the control food. And you can see that these are features that we will be particularly interested in because they increased and over time following consumption of the food here, whereas in the controlled food, they remained pretty stable and didn't change at all. And we have many markers that change in both foods. So these are markers that are just not specific enough for the particular food of interest. So they change in our control food as well. So once we had our series of markers that we're interested in, then of course we had to try identify them, which is can be very difficult. And in particular in the area of food intake biomarkers, it's very difficult because many of the databases don't have the type of biomarkers that we're particularly interested in. So we performed LCMS analysis to try identify and then confirm them where we could with standards, authentic standards. So in particular, I'm going to just focus in on this one, this 2-IPMA, isopropyl malic acid, which we confirmed with a standard. So now we have a putative biomarker, and we had others as well from our intervention study, so our acute intervention study. And now we wanted to move a little bit towards validating this particular biomarker and seeing, well, is it really a biomarker of intake? So we performed a dose response study where we had volunteers eating different portions of the legumes, so a low portion, a medium portion, and a high portion. And we collected urine samples at the end of the week once they had consumed these portions of food in conjunction with their habitual diet over a period. And over here, you can see that this biomarker, for example, increased following increasing consumption of the food. So here we have a biomarker that responded in an acute intervention and also had a dose response to the actual food itself. And then finally, because we were part of this food ball, we were able to get access to some other data from our colleagues. So this is data from the University of Copenhagen, where they had done a very similar study, but they were interested in beef and pork and chicken. But as their control, they had used pea burgers, so legume burgers. So we asked, well, could we see your data? And could we try find our markers of pea in your control food? And lo and behold, here is our marker. And you can see that it increased following in their control food, but actually their test foods, so their meat foods, it remained pretty stable across the time period. So a very nice validation of our biomarker in a completely different set of volunteers. So that was just to give you an idea of how we go about identifying these biomarkers. And there are many different examples in the literature of where people have identified biomarkers of certain foods. And I just give you a snapshot of some of them here. And in particular, I will highlight these ones with the red on top, which are systematic reviews that we've done as part of Food Ball, which looking at biomarkers of different food groups. And we covered a huge range of food groups and really done in detailed literature review and finding biomarkers that have been already associated with these different foods. So I'd encourage you, if you're interested in these, in biomarkers of food intake, to look into these. And down here is an example of a biomarker associated with banana intake, and up here are biomarkers associated with meat and fish. But as I said, lots of examples of people doing very exciting work looking for new biomarkers of intake, and we're getting more and more of them every day. But what we're really interested is, well, yes, it's exciting to find these biomarkers and to do the metabolomics work on it, but how are we actually going to use them? How are we going to move on then from just finding them, document them? How can we move on to use them? As I said, we're interested in trying to use them to determine intake, and our idea or the concept would be that wouldn't it be wonderful if you could take a urine sample, determine a biomarker concentration in that urine sample, and from that understand the food intake in terms of grams per day. So this is, I guess, where we'd like to move towards, and we tested this out and developed some data that you can get towards that level of detail using these biomarkers if you do the correct types of studies to support it. So we started out with a biomarker that was well known from the literature. It was proline betaine, and we quantified proline betaine in a range of different studies. And we also carried out an intervention study in a very controlled environment where we had individuals consume different amounts of orange. And we could see a very nice linear relationship. As you increase the consumption of orange, you increase the excretion of proline betaine. And the concept behind this then was that you could take any urine sample, and from that urine sample, determine the proline betaine concentration of it, and determine using your curves that you've developed, then the actual orange intake or the citrus intake. So we wanted to see, well, actually, could you do this? And could you do this in a population study? And how good would it compare to our gold standard? And our gold standard at the moment for assessing intake is our self-reported data. And the gold standard within that would be food diaries. So we set out about looking to see, could we determine from a urine sample intake of citrus, and how well would it compare to current methods? And the current method we used was a 4-day semi-weighed food diary, which was used to develop or to estimate habitual intake in terms of grams per day. So we wanted the biomarker data, we would plug it into our calibration curves that we developed in the feeding study, and we would estimate intake from that. So to do this, we turned to our National Adult Nutrition Survey in Ireland. This is a food consumption survey, which is a representation of the adult population in Ireland, and it collects detailed dietary data from this population group. And it also collected urine samples. So from the dietary data, and it was semi-weighed food diaries, we were able to estimate citrus intake. So this is what we call our reported citrus intake. And from the urine samples, we measured proline betaine intake in these urine samples. We plugged them into our calibration equations, and we got a predicted citrus intake in terms of grams per day. And we wanted to see, well, how well did this compare to what people were reporting in this particular survey. So before we do that, I want to take one step back, and I'll go back to our intervention study. So now we had a relatively large intervention study, which means that we could actually develop the calibration curves with just 2/3 of the data and leave out 1/3 of the data, just to see in that 1/3 of the data, if we tried to estimate intake, how well it would compare with actual intake, because our controlled intervention study was done in a really controlled environment where individuals were staying in a metabolic suite. So we knew exactly what they were eating. So there was no need to estimate intakes, no self-reporting. We were giving them food and they were consuming that food. And if there was any leftovers, we were able to estimate that the leftovers from in that as well. So we knew exactly what they were eating. So how well did the biomarker data do in estimating what people were consuming in terms of citrus intake? And here you can see this is a snapshot of the data. This is actual orange intake here, and this is what we predicted using the biomarker data. And you can see that it's done pretty well across a range of intakes. So at the low intakes and also at the higher level of intakes, there was pretty good agreement between it. And if you do a very simple correlation analysis between them, between actual orange intake and what we were able to predict using the biomarker data, you get really high correlations. And this is what we got in the 24-hour urine samples. But interestingly, we were able to get as high a correlation just from using your fasting urine samples. And I must say that all these samples are normalized to osmolality. So we take osmolality into consideration when we're doing this. So very exciting for us. We were very excited to see this, that our biomarker data was actually able to tell us what people were eating. So then back to our cross-sectional, our free living, because I guess this is the proof of the pudding, because the controlled environment is extremely controlled. We know exactly what they're eating, but that's not real life. So what happens then in real life? So what we wanted to do in our population study was quantify the proline betaine, plug it into our calibration curves, and get an estimation of citrus intake, which I've called here our predicted citrus intake. And here's a snapshot of the data. This is the recorded intake by the individuals using the 4-day semi-weighed food diary. And here is what we estimate from their urine samples. And again, the first thing you notice is that across a range of intakes, we've done pretty good. So if they were having low recorded intakes, we were estimating low, and when they were higher, we were also estimating higher. So we were very happy with this. And then if you look across the total population, and we've done a Bland-Altman plot to do this, you can look at the agreement between the two methods. And you can see here that the actual overall bias, so that's the difference between the methods, was low. So very good agreement. There are individuals, and you'll see them here, that we ended up with really quite different intakes from what they recorded and what we estimated using the biomarker data. And if you think about it, this is 600 individuals, and you would expect out of those 600 individuals that you would have individuals that are underreporting their food intake. So this is a known phenomenon in nutrition data. And actually, these individuals, what was happening here is that we were estimating a much higher intake than what they were recording. So they were really underreporting what they were eating. So a very nice example that, yes, this biomarker data, these biomarkers can really tell us what people are eating. And if you have the correct controlled intervention studies and you take osmolality into account, you can estimate what people were eating. And you have good agreement to our gold standard. So if we think about validation then, and I just want to touch on validation of these biomarkers because it's really important. And again, we've done a lot of work in this foodball consortium trying to think about a validation scheme and how you would validate these biomarkers. So this was what we came up with, and the work was led by Lars Dragstedt. And we came up with 8 criteria that you should look at when you're looking at biomarkers of food intake. And the first one is pretty obvious. It's plausibility. Does it make sense? Is there a biological reason why this biomarker would be in your urine and it's representing the food intake? The time response, so that's like the kinetics that I showed to you in terms of the legume. Is there a kinetic response? Can you see that once you consume the food that you're getting an increase in excretion of the biomarker? Is there a dose response? This is really important, and it's really important if we're trying to use it in a nutrition context, because we are interested in quantity of intake. How robust are the biomarkers? Are there other confounding foods? Is it specific for that particular food? That's really important. And how does the biomarker estimation compare with other dietary assessment methods? So that's also important to do. And then, of course, you have your analytics. So is the biomarker stable? Is it going to degrade in the biofluid? And then how does the actual measurement of it perform from a performance point of view and also from a reproducibility point of view? So we do try to address all of these when we're proposing and when we're looking at biomarkers. And I think more work at an international level is really needed at this to try push the field forward into getting valid biomarkers of intake. So as I said, we're particularly interested in trying to see how we can use these biomarkers. And I've just given you a very nice example of citrus and proline beetling being a well-established biomarker already in the literature before we started working on it of citrus intake. And what we did was we really just showed or developed methods to show that it can actually tell us about the quantity of citrus intake, and we can estimate citrus intake from it. But then how do you start to use this in epidemiology and any of the biomarkers in epidemiology, where you may have huge amounts of data in terms of a population? So you're talking about maybe 100,000 cohort, etc. Do you need to measure the biomarker data and the food intake biomarker data in all of those subjects? So our idea here was that you potentially don't, especially if you're moving into large epidemiology studies where you have dietary data from those subjects already in terms of food frequency questionnaires, which are relatively cheap to measure. So it will be hard to justify having to measure biomarker intake measures on all subjects. So what we did was we looked into trying to see, could we develop a method or a way of combining biomarker data and our self-reported data? And we borrowed this concept from the nutrition literature, which does something similar for estimating energy intake. So what we've done here was you take your biomarker data and you take your self-reported data on a certain portion of the population group. You model that relationship between them, and then you use the estimates in your model to correct for the error that's going to be in your dietary data in the larger population group. So we done this, and again we use citrus intake as our model, and you can see here that we developed calibration equations where you have your biomarker derived estimates of citrus intake, and you have over here your mean daily self-reported estimate of citrus intake, and you can model these, and then you can use these estimates then to calibrate your daily citrus intake from your biomarker, from your self-reported data in the remaining of the population group. So for example, you could have data, biomarker data on 30 or 40% of the population group, model it, use the estimates then to correct the error that's in the dietary data in the rest of the data set. So we developed an app that's freely available to look at, to download and to play around with. It's called BioIntake. And again, you can see here that we carried out a study just to look at doing this on citrus data, and we modeled the relationship in 600 individuals, and then we used it to correct those estimates, to correct the data, the self-reported data in over 800 individuals, and that's what you see here. So the black is the self-reported data, and the grey is the predicted or the corrected data. So this was, I guess, a nice example of how you would use biomarker data together with self-reported data. But the question then arose, well, how much biomarker data would you need to do this? How could we estimate it? So we've done a number of simulation studies trying to figure this out. And what we could see was that the standard deviations, and you can see here, tend to stabilize quickly when you have around 20 to 30% of the population, regardless of the scenario that we used or we considered. And a quick stabilization means that the collecting biomarker data for the whole population isn't adding any more stability in terms of the calibration equation estimation. So really in this instance, what we could estimate was for this particular biomarker was that about having biomarker data in around 20 to 30% of the population was sufficient. So just to recap on that, so what that concept is that you have your biomarker data, you have your self-reported data, you model both of them in a smaller cohort of your larger population group. You develop calibration equations and you use those calibration equations to correct the larger self-reported data in a larger sample size. And then you have a large sample size of which you have potentially more accurate dietary data. And then you can start to look at the relationship between diet and health outcomes in that large population group. So that's all working, I guess, from the concept of having one biomarker and one food. But what we and others have started to see is that actually most of the foods have more than one biomarker, and this concept of panels of biomarkers has started to emerge. And I think this is going to be more and more important as the field moves forward. So not every biomarker needs to be completely specific, but if it's part of a panel, it could add information into that panel in relation to a particular food. So we were interested then, well, what happens in our case here where you're modeling one biomarker versus one food? How do we model multiple biomarkers against a continuous intake of food? So to do that, we worked with our colleagues in the statistics department in UCD, and we developed a new framework which we called multi-marker, and we developed a corresponding app to go with it, which will allow you to model multiple biomarkers against one particular food, and then to use those estimates for another sample where you had those multiple biomarkers to estimate food intake. And you can see here that you get not just the estimate of food intake, you actually also get a very nice predictive distribution around it. And so you can see how good your prediction is. So as I said, we developed this app here, which you can see here, which is when we called it multi-marker to do this. And the concept is you have multiple biomarkers against a continuous food intake. You model those, you get your model estimations, and then you can predict your food intake in other samples once you have measurement of those biomarkers. And again, that's freely available, and we'd be delighted if people would look into it and start to use it. So then, and the final thing I want to touch on in relation to food intake is the fact that actually we don't just eat single foods. We eat patterns of foods, and you'll all know that. You've eaten your breakfast this morning and it would have been a pattern of intake. You'll have your lunch and then you'll have your dinner. So from a health point of view, we're often very interested in looking at people's dietary patterns and trying to take in a more holistic view of what people are eating. So then from a metabolomic point of view and a biomarker point of view, we were particularly interested in seeing, well, could we develop a model where we took combinations of different biomarkers, and then we could relatively quickly classify an individual into a dietary pattern using that model. So we started out with a very crude model where we could classify individuals either into a healthy or unhealthy dietary pattern, but we then refined that model more recently. And what we have here now is a model where we were able to build a model and classify based on metabolomic and biomarker data and classify people into 4 different dietary patterns. And what we wanted to do then was say, if we took an independent study, could we replicate those dietary patterns and were they stable over time? And could we classify individuals into the same dietary pattern over time using the metabolite data? So what were the 4 dietary patterns? So we identified 4 dietary patterns. As I said, one is a moderately unhealthy. The second is an unhealthy dietary pattern, moderately healthy, and then the prudent is the healthiest of the 4 dietary patterns. And you can see that very nicely here. These are diet quality scores that we often use in nutrition to indicate the overall quality of the diet. So the first one is the Alternative Healthy Eating Index, and the second one is a Mediterranean diet. And you can see that they're both higher for the prudent diet and they're lowest for the unhealthy diet. And you can see that there are other food groups here that are, for example, if you look down here, you will see the high energy beverages. So these are more like sugary drinks would be lowest in the prudent diet, and then the unhealthy diet, the intake of these would be highest. So then we also looked at nutrient status, and were our dietary patterns backed up by having nutrient status in these individuals. So we measured nutrients and biochemical markers in blood samples from these individuals. And again, you can see here, this is cluster 4. So this was our healthy cluster. You can see it has the highest circulating levels of the B vitamin folate, for example, and it has the lowest excretion of urinary sodium as well, whereas our unhealthy diet had the lowest level of this particular biomarker as an example, and it had the highest level of excretion of sodium in the urine. So a nice validation that we did have actually four different dietary patterns, both from the food, the dietary data, and also from the biomarker data in the blood. And these dietary patterns were developed, just to re-emphasize that, from our models, from our biomarker models, based on metabolic profiles of the urine samples. So what we wanted to do then was to see, could we reproduce this in an independent study, and how reproducible was the classification of the biomarkers? So if somebody was classified as in an unhealthy dietary pattern, was that reproducible over time? So you can see that here for two of the 4 clusters. So this was the prudent and this was the moderately healthy dietary cluster. And you can see again, we had 4 visits for these individuals, 1 visit once a month. So this is over a 4-month period. And you can see from these plots that you have very reproducible dietary patterns in these individuals. So the take-home message is that we were able to build a model using the biomarker data, classify individuals into dietary patterns, and we validated these dietary patterns using nutrient data. And then more importantly, we were able to replicate these in an independent study, and that the classification was reproducible over time. So I hope that I've shown you that biomarkers can tell us about food intake. And they can be very, very informative in that. But not all biomarkers are going to give us a linear relationship with food, or they're not going to give us that nice relationship you saw with citrus intake. And we wouldn't expect them to do it either. But that doesn't mean that they don't contain useful information from a food point of view, as long as we acknowledge that we're not going to get quantitative food intake data. You might have a biomarker that's influenced by food intake, but actually that it's also influenced by other factors such as the microbiome and the host genetics. And that biomarker could be very valuable as well. And it could be very valuable in looking at the relationship with health parameters, disease risk factors, and disease outcome measures. And diet is just one of the factors that are influencing that biomarker. And again, there are very nice examples in the literature where this type of approach for these types of biomarkers are coming into play. And again, in the interest of time, I don't have time to go into any of these examples today, but just to say that just because a biomarker doesn't have a complete beautiful dose response with a food intake doesn't mean that it's not useful. It can be useful in a different context, not telling us exactly what food people have eaten, but actually in studying the relationship between health and disease. So then if I move on then to the last part of my presentation, which I wanted to move into the concept of metabolic phenotyping or precision nutrition. And the context behind all of this is that over here on our left, we have this one-size-fits-all approach to dietary advice, where everybody gets the same advice. And we know that this is not good enough anymore, that we need to move away from this approach, and we need to move more towards a personalized approach. And the personalized approach is really here, that you have an advice tailored to an individual. But we've been interested for many years now in trying to see, well, actually, are there subgroups or clusters of individuals in the population that we could group together that we know are metabolically very similar, and then we could tailor the advice to these individuals based on their cluster or their subgrouping. So this we have used it in the context of doing metabotyping, but really from a nutrition perspective, in trying to figure out if we could tailor the dietary advice to these individuals. And we've published a number of papers where we've been interested in developing these concepts. But to go into the concept in a little bit more in detail, so essentially the concept is that you take your metabolic data, you perform a cluster analysis, and it can be any type of clustering analysis. One that we often use is k-means. You group your individuals based on their metabolic profile into their different groups, and then you build on that some of their own characteristics, and you can develop decision trees and automated decision trees to do this. So you can add in BMI, you can add in waist circumference, you can add in blood pressure, and you can then deliver targeted advice to that individual based on the fact that they're a member of a specific metabotype, but also some of their own individual characteristics. So we developed a framework from the Irish National Nutrition Survey to do this. So using our 896 adults in this survey, we developed 3 metabotypes that we could identify in the group. Metabotype 1 had normal levels of triglycerides, HDL and glucose, but it had a slightly elevated total cholesterol. Metabotype 2 was our healthy metabotype, and metabotype 3 then had high levels of triglycerides, glycerols, total cholesterol and glucose. And for each of these metabotypes, we developed a series of decision trees. And we developed this first, and then more recently, we have automated this and updated this with the latest evidence and guidelines. And when we do an analysis on this retrospectively on cohorts, we can see that we can pick out metabotypes, we can give them dietary advice using these decision trees, and we have a very good accuracy if we compare it to what somebody would do if they sat down with a person and done it on an individual level basis. So, but what we've done more recently is that, well, actually, if we try to develop a metabotype approach, can we actually deliver dietary advice to the individuals better than what you would in a standard one-for-all approach? So we've just finished a randomized control single-blind trial where we included individuals, men and women, aged between 18 and 65 and BMI greater than 18.5, where our primary outcome was a change in diet quality, and our secondary outcome was change in blood biochemistry. And our hypothesis here was that if we give individuals personalized advice based on their metabotype, they would change their diet quality more than what they would if they received personalized dietary advice in the control group. So we had randomization into what we call our personalized group, and in our personalized group then we had our 3 metabotypes, and we give them dietary advice based on their metabolic phenotype or their metabotype. And then our control group just got our standard dietary advice. And again, we're just crunching the numbers on this, and it's looking good, but I can't tell you the results because we're not finished crunching numbers yet. But I hope the next time you hear me speak, we'll be able to present some of this data, and we hope we'll be able to do that during the summer. So then I just want to spend the last few minutes wrapping up and talking about maybe some conclusions and challenges for the field, and where do we go forward. So in the context of metabolomics, there's without a doubt, in the context of human nutrition, metabolomics has a huge role to play. It's really beginning to be realized now at the moment, and I think we have a very exciting 5 to 10 years ahead of us as metabolomic applications become more and more in human nutrition research. In the context of food intake biomarkers, I've shown you some data where the biomarker data performed as well as our current gold standard, which is our 4-day semi-weighed food diaries. We believe that there are many more markers that will perform very well in terms of food intake, and that there will be foods that we will be able to estimate intake using biomarker approaches. Biomarkers then has a huge potential in the classification of people into dietary patterns, and that's again huge in the context of studying of epidemiology. So if you can quickly use your biomarker data to classify an individual into a dietary pattern, then you can look to see what is the impact of that dietary pattern over time. And again, in the context of delivering personalized nutrition, if you can classify quickly a person into a dietary pattern and give them advice based on that, then that has huge potential as well. And in the area of precision nutrition, I touched on some of this, metabolomics in the context of metabolic phenotyping, and trying to give advice back to individuals based on their metabolic phenotype is playing a key role, and some very interesting studies coming out in that context. However, it might all seem very easy, but that's not the case. We have huge challenges as we move forward. And I think this first challenge that I put up here is a huge issue for us. We have many unassigned features in our profiles. We know that they're related to food intake, but the databases just don't have the metabolites in there. In many instances, if we're trying to confirm the identification, we have to rely on getting the marker that we think it is synthesized, and it can be a very slow and arduous process. So I think that this area really needs alignment at an international level, and we need to join forces to try and improve these identification of these unassigned features from our perspective anyway, in terms of food biomarkers. There are huge numbers of food biomarkers out there in the literature that just haven't been validated yet. The validation work is difficult. It's probably not the most exciting sometimes to do, but it's actually really important for downstream use of the biomarker, that it is a valid biomarker. So this area, again, I would flag as a really important area that we need to pay attention to. The area of dietary patterns and having biomarkers and biomarker profiles help classify individuals into a dietary patterns is an area that's really ripe for further development. I've shown you data where we just did it for 4 dietary patterns, but of course there are much more dietary patterns and more sophisticated tools that we could use to develop that further. A question that's often asked is then, well, how many, if you're going to use biomarkers, how many repeated measures should you use to get a good capture of an individual's dietary intake? So from a nutrition perspective, we would use a semi-weighed 4-day food diary. So we would be looking over 4 days to see what the habitual intake of that person is. So again, we should be taking repeated measures, samples from individuals from a urine point of view and blood, if we're going to look for biomarkers of intake, and really if we want those biomarkers to reflect their habitual intake. And I touched a bit on combining food intake with dietary data. I give you one example. We're trying to work with other examples at the moment, because this again is an area that has huge potential in trying to scale up what we're doing and trying to really have an impact on nutrition epidemiology. And not to finish on the challenges, let's finish on the opportunities. There's huge opportunities in the area. As I said, intake determination is a huge challenge for nutrition research, and I think metabolomics can help us play a role. It can tell us for actual intake, but actually there will be lots of biomarkers that won't work very well and won't be able to give us the precise intake, and they might work just at a classification level, and that's okay. So trying to classify individuals into low, medium, and high. So that's probably as good as what we can do sometimes from our self-reported data. So I think bio intake again needs a lot of development and huge potential here. Intermediate biomarkers, so these are what I was alluding to. So these are biomarkers that are reflect food intake, but are actually also influenced by other processes. So it could be the microbiome, it could be genetics, it could be physical exercise, etc. So these biomarkers again won't tell us about food intake in a quantitative fashion, but they can tell us some very useful information. Dietary patterns, I think again we have a huge amount of work to do on that, and then we're only starting to see the benefits of metabolomics in precision nutrition. So with that, before I finish up, I'd like to acknowledge the team in UCD. I'm very privileged to work with a fantastic group of individuals. I also have some fabulous collaborators across Europe and in Ireland. And I'd like to also acknowledge the range of funders we have for funding the lab. And thank you for listening, and I look forward to answering any questions.
Thank you so much, Dr. Brennan. That was a fantastic overview and really good insights in how this is established and how it correlates to self-reported, and how it can be validated. It's really great to have this inclusive way. Arpana, why don't you lead the discussion?
Great. Thank you, Dr. Brennan. That was a wonderful talk. Just jumping right into the citrus intake study, very interesting stuff. There was a question, do you get equally nice correlations for proline betaine if you normalize urine concentrations to creatinine instead of osmolality?
For this particular study, we didn't use creatinine. We tend to use, we use osmolality. It has worked better for us than creatinine. We find that creatinine can be influenced quite a lot by gender and also by physical activity. So osmolality has worked better for us.
Great, thank you. In human cohort studies, we have many more samples with plasma samples than with urine. Have you taken on blood-based food biomarkers in your research?
Yes, so we have the examples I've shown you now to date today were predominantly from urine work. We have done work in blood as well. Blood will be particularly important, for example, for lipids and lipid foods that have high lipid content. Blood will also be important for markers maybe that have longer half-lives in the body. So depends on the food, I'm afraid, is the answer, and it depends on the type of markers that are in that particular food. So I wouldn't rule out blood completely. I think it is useful, and probably it will be a combination of blood and urine.
Interesting. I never realized that, it depends on food, and then like the sample type, it impacts that. Hadn't thought about that. Going along the same study here, it was really interesting to see how well your prediction model predicted those values. So in that current prediction model, can you differentiate consumption of fresh orange juice perhaps and compared to fresh juice from concentrate? That's a very interesting question.
We didn't look into that, but we were only interested in total citrus intake, and then again we just had total citrus intake, which would have included both in our food diary data.
Okay, would that even matter in terms of like where the research and the field is going in terms of differentiating to that certain level, whether it's like fresh orange or juice?
So it depends on them. It depends on the food. So there's some instances it might be interesting to look at whether it's a fresh food you're eating or whether it's a concentrated food that you're having, or whether it's a processed food or not. In that instance, that's probably there is an interest in looking for biomarkers that potentially for certain foods could differentiate those. But actually, you could probably also use your self-reported data to help you with that a little bit. So and that's the kind of beauty as such as having that self-reported data. It can help you disentangle some of those things that maybe the biomarkers can't.
Okay, thank you. There was a question here, and I think it's just based, very general question about food intake. Since nutrients in food influences absorption and metabolism, how do you resolve such interactions while estimating a collection of food intakes?
So nutrients. So we're in a collection of food intakes. And I'm not sure I fully understand the question, but I'll give it a shot.
I think what they're asking is just how do you resolve interactions when it comes to other nutrients in foods, especially when there's an influence between absorption and metabolism of these nutrients alongside with other biomarkers.
Okay, so I guess we take it back to how we do the discovery studies. So the discovery studies are often done with one food. So you're feeding a particular food. So you'll find a biomarker that's associated with that food. Now, if that biomarker is going to be influenced by other foods, you'll find you probably won't see that so much in your, you should actually, if your dose response is done in the background of a habitual diet, that biomarker won't perform very well in that dose response study, because you will have other foods consumed there as well, and you'll be looking to see, is it increasing as portions of food increase. So you'll probably get a flag there that your biomarker isn't working or is being influenced by other foods and other processes. And then finally, if you move into a cross-sectional study where you have people eating everything and anything, and your biomarker is probably won't hold up in that context either. So there's a couple of ways along the route, I guess, of validating the biomarkers that will help you to identify whether you have an issue with specificity, I guess, is the word in the context of a food intake, or whether it's being influenced by other biomarkers along the way. And that biomarker then might potentially just be useful as a part of a panel of biomarkers, but not on its own.
Thank you. I think there's another question that's kind of on the similar track as what we just talked about. So in terms of utilizing food biomarkers to predict dietary intake, what are your thoughts on key factors that potentially explain small differences between predicted versus actual intake?
So, but on small differences between predicted and actual intake?
Yeah, so there was a couple examples here. They had said, for example, inter-individual variability in absorption, digestion, and metabolism, or baseline status differences.
Yeah, so if you're going to have a huge inter-individual variation on the biomarker, so if it's going to vary hugely between your individuals, again, you'll see that in your data at them, probably already at the acute study side, where you'll see whether or not your kinetics is hugely variable across your individuals. Now, if a biomarker is so variable and has very large inter-individual variation across, we'll say in the kinetics part of it, it's probably, when you move it into a population study, probably not going to give you accurate data from a food intake point of view. It might be quite difficult. So you'll have that flag, I think, there. And then again, when you go into the dose response, what you might see is a huge variation as you change the portion of the food. And again, you can have a look to see, am I getting at this higher variation as the intake of the food increases, for example. So you can tease out some of that data there. You could actually potentially use some of that data and to help you as well. Well, actually, is there subgroups of individuals that this biomarker works very well for? Or can I include some of these covariates that are in my models and in my equations? And then you're modeling them with your biomarker data as well.
Great, thank you. I had a question going back to the study with the peas, with the food ball project. You reported one biomarker with a level 1 identification. When you're trying to expand and trying to find multiple biomarkers in the project that you were talking about in later in your talk, what levels are you reporting those biomarkers? Is it always level 1, level 2? What is kind of like the gold standard in nutrition metabolomics?
Well, we try to go for level 1 if we can, but as I said towards the end of my talk, that can be difficult, just purely by the fact that we don't have a lot of these compounds, and we have to end up synthesizing them if we need to validate them completely. So yes, there would be a drive to try to be at level 1 at least, if not then level 2.
Great, thank you. And something that is equally important, there's a great question here. What's the impact of the microbiome on the dietary biomarkers? Microbiome is such a hot topic and very relevant, I guess, when it comes to nutrition.
Yeah, absolutely. So the microbiome, as I've said in one of the slides, it can have a big influence on some of the biomarkers that you see. Again, trying to figure out whether your biomarker is going to be influenced by that or not. So there are many biomarkers that won't be influenced by the microbiome, and then there will be others that will be influenced by the microbiome, and that will be actually useful information as well. So again, in your acute studies, you can figure out probably quite easily whether you're going to have a biomarker that's going to be influenced or not by the microbiome, just by virtue of the kinetics of it. But then when you come to your dose response studies, you have to be careful and you have to look at them and take that into consideration.
Great, thank you. Switching gears to your precision nutrition portion of your lecture, can you share how you chose k-means and validated clusters for the pre-decision tree process?
How do we choose k-means? It comes from the nutrition field. We would use k-means quite a lot in classifying dietary data. So it works quite well in that context. How we decide on the number of clusters, in this particular study, we used the AIC criteria to identify the number of clusters that we would have, or the number of clusters.
Great, thank you. What would be the influence of different population groups in determining food intake biomarkers and in precision nutrition?
The influence of different population groups, you mean the kind of metabotypes?
Yeah, I would assume that's
so. So in the metabotypes that I was showing, I guess those are based on metabolites that are endogenous metabolites, so they're not biomarkers of food intake. So they're not coming from food. So we distinguish very clearly our exogenous metabolites, or metabolites that we think are coming exogenously, from metabolites that we know that are endogenous. When we're doing our metabotyping, we're focused on our endogenous metabolites, and we're trying to get metabotyping based on that and the metabolic phenotype on metabolic pathways that we have. So that's how we do it. So we're really thinking that we don't want these metabotypes to be influenced by food intake. We want them by food intake biomarkers, I should say, not to be influenced by food intake, but by food intake biomarkers. And we keep very separately, and we have different platforms for running our samples on that, and we keep very separate. Our food intake biomarkers are our exogenous markers. So they're markers that are coming from the food as opposed to metabolites that we would find endogenously produced.
Great. This made me think of another question, and maybe it's because I'm not in the nutrition field, but very much interested in food and natural products, things like that. Do you normally have parallel studies where you just look at the food samples themselves and then compare them to, the samples that you're taking from people in terms of like the urine samples that you talked about, to kind of correlate what you're finding? Is that normally? Okay.
Yeah, that's absolutely. So we would try to go back to our foods. We keep all the foods. We actually keep the foods that we feed to the individuals. And we would usually also get other samples of the foods, and if possible, get them from different countries of origin and different places, just to see how influenced how diverse levels of the biomarkers are present in those different foods. But that has been very, very useful for us actually to go back to the foods and to look at them, the metabolites present in the foods.
Great, thank you. That's what I assumed, but I wanted to hear from the expert. And then there's a question here: for the creation of metabotypes, from your experience, any other approach apart from k-means? I know you said k-means is, foundation, like very much used in nutrition, but are there other clustering approaches? And then based on the definition of different disease risks, I guess, are there other types of correlations used based on the disease risk itself?
Yeah, no, there's lots of different clustering approaches used in metabotypes. I know hierarchical clustering is used. MCLUST is used a lot as well, and there's some Bayesian approaches as well from a clustering point of view. So there are loads of different approaches, and we are looking into some different ones again from our perspective, but at the moment, but we have previously relied heavily on k-means.
Great, thank you. I know there's a question here in the chat about multi-omics. What is the potential of multi-omics for your research? Also, is there any potential of using other machine learning methods instead of what was presented in terms of clustering and decision trees?
Yeah, I mean, absolutely, there will be loads of different machine learning approaches that could be used to learn from it. In particular, I think, what was I going to say? I'm just looking at some of the questions, trying to understand them coming in. So in particular, what I think has been used in the I lost my train of thought now. In the nutrition field would have been, well, as I said, would have been predominantly k-means, but there's another one that's been used a lot now. I can't remember the name of it, but it's an area that's absolutely ripe for development in terms of using different data sets and trying to combine the dietary data, which is actually really, really complex data to work with, combining it with other omics data sets. We haven't developed any particular tools for that multi-omic integration or anything yet.
Yeah, that's definitely a difficult part in terms of integrating multi-omics data. Definitely a challenge there. There's a last question here in the chat. Any thoughts on how to cope with different kinetics of appearance of metabolites in urine or plasma and their use as biomarkers of intake?
Yeah, so I guess that's the time dependent. So you might have a variation in that depending on an individual, and an individual might have a lower level of a biomarker. If there's a huge variation in that, again, your acute study will show it to you, and so you'll be able to flag it from the beginning. But that's something that actually the self-reported data could help you with, because if they've reported consumption of it and you're seeing lower than what you would expect values of it, you could help use your self-reported data to help you there. If again you can understand the kinetics and the variation in the kinetics, and if you can see that there are covariates that explain or influence the kinetics, you can include those kinetics then in your model. So not those kinetics, those covariates in your model. So for example, you might want to include if BMI was having a major impact or age was having a major impact. You can add these into your equations and model them and include them in it, and model the impact it's having on your kinetics excretion.
That's great. I see Oliver's ready for a question here.
No. I just want to invite anyone who feels that they have still questions open, that they feel invited to go directly and talk to us.
Yeah, there's definitely time for that. Those were some of the last questions in the chat, and I was going to say the same thing. If you have questions, please feel free to unmute yourself, or keep posting them in the chat. I see a few people with their cameras turned on, so maybe you're ready to ask a question to Dr. Brennan.
All right, then if that's not the case, then I would like to thank Dr. Brennan again for a fantastic talk. As always, we have learned a lot, and it was very solid. It's very, very also similar to what we try to do here at UCD, with at the real UCD, of course. But on a small scale, of course, right? We don't do very large studies. It's not comparable to multi-year European consortia, but we do it on a small study. Although the US has also embarked on very large studies in precision nutrition, just recently. We'll see what comes out of that. Where UC Davis is also a part of the recruitment and dietary assessment. So it's going to be interesting. So Arpana, maybe you want to tell us about our next month's meeting?
Yes, of course. Let me share my screen here. And we have Dr. Justin Vanderhooft coming on March 30th. He's an assistant professor at Wageningen University and a professor of computational metabolomics. His lab is developing algorithms and models to improve structural annotations of metabolite features. So definitely something we discussed today and in our lecture seminar today. And Dr. Vanderhooft is actually a pioneer of using word and embedding NLP approaches to aid in these analysis. So definitely looking forward to hearing that and hearing his talk on his research.
Okay. With that, thank you again, Dr. Brennan. Thanks everyone for joining. We will upload the recordings soon on our YouTube channel. And we hope that we meet you again next month.
Great.
Bye-bye, everyone.
Thank you. Bye-bye.
Bye-bye.